Can you guys all see my screen?
It says loading.
It says
Yeah, we can, but it's still black.
Okay. Can you see me now?
There it is.
Okay, great. All right, so today we're going to cover some cool CDCN news that I will share. And then I'm going to turn things over to two different team members who are going to share some really awesome research updates, things that they're currently working on. And then I'll turn things over to Anya, who you guys all, I believe, know. She's our biomedical leadership fellow, but also fearless leader for all things roadmap. And she's going to share some cool updates with you guys on that. And then as usual, we'll end it with an Ask Us Anything. And that's where all of our team, David, myself, Anya, and the people who presented will be there just to answer any questions that you guys have, either about the content that we covered today or just in general, things that are going on with you, with Castleman disease and things like that. These are the people who will be speaking tonight. So, you know, David and myself. I did mention Anya. Mike and Melanie are two of our researcher leads at the Castle. They work every single day in the lab and on their computers, combing through all kinds of data that I don't even understand. I still won't understand it after tonight when they do give you the updates, but it's super important things that continue to drive forward our life-saving research. We definitely could not make the advances that we're making without them. So it'll be really exciting to hear what they have to say. So why are we here? Rare Disease Day. It is a day where every single person in the rare disease community uses this to raise awareness for their specific rare disease. So I just really kind of wanted to touch on, you know, overall what Rare Disease Day is. There are 300 million people worldwide living with some type of rare disease. There's over 6,000 different rare diseases that have been identified. So for us personally, we use this day to raise awareness, to share stories, to communicate updates on what the CDCN and the Castle are doing. I personally love being able to share why we do this, and that's because of all of you. So we sit here doing the work that we're doing every single day so that all of our patients, their loved ones, their families can continue to live the fullest life they possibly can. I love seeing all these faces that people have you know, shared photos with me over the last, you know, year or so, just of how they're doing and give me updates. And on our Rare Disease Day, we love to share these stories and share your colors. And so for Castleman disease, it's just all about us bringing more awareness to it and kind of sharing with you guys in a community gathering the different things that we're doing. If all of you guys are on Facebook and saw some of the posts that we've been making, we used the Rare Disease Day and turned it into Rare Disease Month, where we were kind of pushing to try to obtain 10 blood samples over the course of this month. I'm really happy to share that not only did we achieve that goal of 10 blood samples, but we actually brought in 14 donations this month. That is wonderful. As I'm sure you've heard us say millions and millions of times over the last decade, we cannot do the work that we're doing in the lab without our patients donating blood, donating tissue, enrolling it into our natural history registry. There's such vital, important steps that you guys as patients can do to continue to drive forward this research. When we are able to achieve something like 14 in 1 month, which is the shortest month in the year, it was really exciting. So we celebrated that on our morning check-in this morning. And then I just wanted to make sure to communicate to you guys, we're so thankful for what you guys do to contribute to the CDCN, to join in the fight. And it was just, it was a really great thing to see that we hit that goal. So thank you guys. Before I advance to the next slide, because I'm going to ruin it if I push too soon, the next slide is a video that I'm going to share with you guys. Jimmy, who is the spouse to Mary Zaccato, our executive director, he put together an amazing video on our patient summit last year. This video is what we're going to use to continue to promote the awesome work that we do at Summit, the importance of what Summit is for our community, but it also is going to announce to you when the next date is. So pay attention. And here we go.
When we think about the Patient Summit, we always say that this is our Super Bowl. Super Bowl is the biggest weekend of the year, when sort of all of your training and all of your hard work comes together and you have this opportunity to do everything you can. And that's really what this weekend is for us with the CDCN. We get patients together with our amazing researchers, with our team from the CDCN, and we get to talk about Castleman disease. We get to talk about the research that we've been doing. We get to forge forward a path for how we're going to continue to push forward against Castleman disease over the next decade. I co-founded the CDCN with Dr. Fritz Van Ree, who's here today.
I think it's been an amazing journey. A lot has been accomplished. We've brought together patients, their families, physicians, scientists. We discovered new drugs. We developed treatment guidelines, diagnostic guidelines.
The goal and the vision is a world where every patient diagnosed with Castleman disease receives a therapy so they can live a full life.
So big news, I am already working alongside members of our team to plan the next Patient and Loved One Summit. So save that date. I know a lot of you have already been asking me. Actually, everybody started asking. The second summit ended last year. So it is August 10th and 11th. It will definitely have the hybrid option again. So for those of you who cannot attend in person here in Philadelphia, we will definitely have links sent out to attend via Zoom. More details will come over the next few weeks to months as we get more things planned, but make sure to mark your calendars. We'd love to have all of you here. It's my favorite time of the year because I physically get to hug everybody. I get to see everybody. I get to hear how everybody's doing. And it is a really awesome weekend where you guys can gain all kinds of education on Castleman disease. You get to meet members of our team in person and really just get to connect with each other, which I think is a huge positive aspect of our patient summit. Penny,
Leva,
yeah.
that's going to carry over to the 12th, correct?
Yes.
Or are we not doing Saturday?
It's the whole week. It's that weekend, Thursday, Friday, Saturday.
Okay, Thursday, Friday, Saturday. Okay.
Yep. And another really big news that I wanted to share with you guys is that because of our community, and that's our patients, our loved ones, our physicians, our researchers, our donors, community members, family, friends, we were able to raise almost $500,000 this year, which is amazing. And this does not include, this is just donations from donors, from Quest, fundraising, Facebook, all of you guys, you know, posting for your birthdays, Facebook fundraisers. So it's really, really exciting to share to you guys that we raised almost $500,000 for our research. I want to say continue that throughout this year. I'd love to achieve even more than that. So if I tend to bother you guys and remind you, hey, your birthday's coming up, post a fundraiser, that is just because I know that as a community, when we rally together, we can achieve milestones like this. So super, super exciting. And I just, from the bottom of my heart, thank you guys for all that you continue to do. Now I'm going to turn things over to Michael. He is one of our lead researchers at the CSTL. He spends every single day, like I said, combing through things I do not understand. But I'm going to let him kind of share some really awesome updates on what he is focusing on this year that is super important for our community.
All right. Thanks, Mileva. And very happy to be here. Just a bit about me before I kind of dive into, you know, what we're going to cover today. I'm Mike Gonzalez. I've been with the center for about 1.5 years now. I'm what's called a computational biologist. So I use the processing power of computers to dig into all of the data that you guys provide as far as medical records and samples that we do testing on. And I try to use computers to try and piece together the Castleman puzzle. So I'm coming over from several years as a senior scientist at the Children's Hospital of Philadelphia, where I dug into a number of rare pediatric disorders, and now I'm focusing my efforts on Castleman disease. So excited to be here, excited to share some updates, and I'm particularly excited about what I'm going to share because I think we're going to make some really major progress on these things over the next year. You can go forward. And so the first project I'm particularly excited about is centered around this question: are there genetic changes that lead to Castleman disease, or are there genetic changes that can predispose or make it more likely that you develop Castleman disease? And we do this in a few different ways that I'm going to share with you over the next 2 slides, but I really want to kind of pause here and emphasize that really the engine that makes all of these experiments run are all of your samples that you've provided, all of your information that you've provided to our Accelerate database. And so specific shout out to Bridget Austin, who's on the call, our awesome biobank coordinator. If you are interested in getting involved, getting your samples into any of the studies that I described today, please reach out to her. She's more than happy to get your samples collected and get your samples into these studies. But what we essentially do for these genetic type studies, this particular slide is known as family-based genetic studies. And we extract DNA from the samples that you provide. And remember, DNA is our genetic material. That's a mix of our mom and it's a mix of our dad, and it's present in all of the cells of our body. And we can sort of think of DNA as kind of like a cookbook that makes us. DNA is organized into smaller units called genes, and we can sort of think of these genes as individual recipes within each of those cookbooks that eventually produce us. And what we can do is we can use machines that we have at our disposal that could actually read those recipes called DNA sequencers. And we can use specialized computer programs to tell us where your genes as a Castleman patient is just a little bit different from your parents' genes or from your related sibling or family member. And so by comparing Castleman patient DNA to a parent or related family member that does not have Castleman's, we can start to identify those small genetic changes that could be leading to Castleman disease. Next. And you might be asking yourself, well, that's great if you have, a full family-based study, if you have the mom, the dad, a sibling, but what if you just have my sample? And the answer is we can do quite a bit with just your DNA as a Castleman patient. And we've been collecting these samples for quite some time. We currently have about 150, and we have a small number that have already been sequenced, but we actually use a very similar setup where we can actually use those specialized machines again in the same way to read that recipe book. But instead of comparing it to someone like your mom or dad or sibling, we now compare your DNA to publicly available databases of all of the cookbooks that we have access to see where those specific changes in that recipe occurs that could potentially be leading to Castleman's. And at the end of the day, what does this actually give us? I think by understanding the genetic changes that influence Castleman disease, it can really help us understand not only what's happening during disease flare, but it could eventually help us identify potential drug targets for improved therapy. Next. Now, the second project I'm particularly excited about is investigating genes that are active or what's called expressed in Castleman disease lymph node tissue. And this might sound odd because we just got done talking about DNA. And what I mean when I say what genes are expressed in a particular tissue is that although all of our cells that make up our tissues have all of our genes, so all 20,000 that we currently know of, only about 1/10 of those genes are expressed at any one given time or switched on in a particular cell or tissue. And our cells that are the building blocks of our tissues look and act the way they do because of the specific genes that they express and the amounts of gene products that get produced. And not only,
the red dots, please.
and not only can we measure the amounts of these genes, but we can compare them to control lymph node tissue to tell us what genes to focus on in Castleman disease. And so, for example, if you focus on kind of the center of our figure here, you can see that we would begin to ask questions about gene A and gene D as they're both produced more often in Castleman disease tissue, as opposed to something like gene B that has equal levels, probably not as biologically interesting, gene C not expressed in the tissue. And so gene A and gene D are really our candidates for further exploration. And what you see all the way over on the right are actual results from our experiment here. And I'm not going to dig into it for the sake of time, but we're starting to identify these gene candidates that are expressed more or less often compared to controls, and we're beginning to determine how they fit into the overall Castleman disease puzzle. Next. And finally, sort of the last set of experiments I really wanted to touch on is to describe these experiments that center around the idea of improving the speed and accuracy of Castleman disease diagnosis. I'm sure all of us on this Zoom call right now are very well aware of how the diagnostic process should go, but for a number of reasons is not always the quickest or most accurate. And we're trying to improve this workflow through a number of different mechanisms. And I'll touch on two specific ways here. You can go to the next slide. The first is taking advantage of all of the data that we have saved related to not only your lymph node biopsy images, but also the medical records that you've provided to the Accelerate database. We can use all of this data and apply what's called machine learning, which is a type of artificial intelligence that allows us to use, we have over 1,000 lymph node images now and thousands of medical records that you provided to train an algorithm or a set of instructions for a computer that can take lymph node images that have not been diagnosed and compare it to what it's seen from your images and actually tell the likelihood of a Castleman disease diagnosis. And you can imagine if a doctor, is kind of waffling on whether to give that Castleman disease diagnosis and they see a high score with this machine learning algorithm, they may be prompted to give a diagnosis with increased confidence and speed. And then finally, last but not least, we have started to use extremely novel technologies that allow us to take very deep, detailed looks at your lymph node tissue and determine where specific cell types are located in that tissue and how they might be functioning. And this could really help us in identifying cells within the lymph node that could be causing problems and how those cells are actually functioning within Castleman disease. And down the line could eventually help us develop cell-specific and targeted therapies that might help Castleman's. And with that, I would just like to thank you guys for listening to some of the projects that I am particularly excited about. I will pass it to my colleague, Dr. Melanie Mumau, who is a very talented immunologist and cell biologist that leads up a lot of our wet lab work that we have going on at the center.
Thanks, Mike. Hi, everyone. Nice to virtually meet a lot of you. So I always like to say I'm the yin to Mike's yang, or vice versa. So my background is really in experimental immunology. And so basically I'll talk to you a little bit about how I really focus more on the cellular level, whereas I think Mike focuses a little bit more on the molecular level, but how your cells might be interacting in your bodies, and how that might be promoting the disease when you have a flare, and ultimately to determine new ways that we can treat your disease. So the first slide. So I first wanted to just talk about a little bit, because you hear us talk about, it's great that we got 14 samples this month. So thanks to all of you, and especially Bridget, but what happens when we receive your blood sample? And so after Bridget gets it, we process the cells essentially by, we separate the red blood cells from the white cells, white blood cells by centrifugation, which I'm sure you've heard before, but basically based on, how dense things are, you can separate them into different layers. And so we do this. And so the red blood cells fall to the bottom, the white blood cells create this little layer in the middle. And then at the top is this layer of basically fluid, what we call plasma or serum, which has lots of goodies, to help keep us alive and well. But obviously during a flare, we can identify certain proteins. I'm sure you've heard us say cytokine storm sometimes if you have IMCD, perhaps promoting your disease. But I'm sure a lot of people, just given the days of COVID, have also heard cytokine storms. So they're essentially just proteins that are important for a normal immune function, but during COVID and obviously during certain cases of Castleman disease can really promote flares and a lot of the symptoms that you have. So next slide. And so what do we do with these different layers? And so one of the things that we've done a little bit before and we're continuing to do is just characterize the crap out of everything that we get. So mostly with the plasma and white blood cells. So plasma, as I mentioned, has lots of cytokines and other proteins that we characterize. And so really this is, what we do with, we use flare blood mostly for this, but we just characterize all the different proteins that might be important. And so if we see something that is more represented during flare compared to remission or through a healthy donor, then we could, we characterize that as important. And so one example of this that we've done in the past is you've probably heard us talk about this protein CXCL13. The name doesn't necessarily mean anything other than it's important just for you to know that in IMCD patients, it's upregulated. And so it's not just a protein floating around, it has a function and it binds to certain receptors on white blood cells. So we can take, your sample from flare and at the same time characterize the plasma, but also characterize the white blood cells. And so, we've been able to see differences in the receptor that it binds to. And so, these two pieces of information we're hoping to kind of connect, as well as combining that with clinical data that we learned from Accelerate. So are there other lab values and certain types of things that, we can glean, like I said, from the patient registry and kind of put all of this together and just generate new insights into the disease. So just to summarize, take plasma, characterize that, we characterize all the white blood cells, and we take the clinical data, put it all together, and hopefully we can generate some new findings. And then next slide. And the second thing that I just wanted to talk about, so we have learned, like I said, a little bit about what's in the plasma and what types of blood cells might be important, but we also want to try to model this in a dish, essentially. And so one of the things that we're actively doing right now is actually taking CXCL13 and throwing that onto cells that will respond to it, and then basically seeing what happens. So do those cells grow and proliferate? Do they make more of them? And perhaps they secrete other cytokines or other proteins that might be promoting flare. And also, they might be activating cell processes. So I'm sure many of you are aware that, we know that mTOR, for example, is important for IMCD. And so, is this, can we discover new things that ultimately can be targeted by drugs? And so these are three just examples of things that we could potentially target. And I wanted to just mention that this is, we model basically flare in a dish, and we do this with your remission samples. So both flare and remission samples are important. And these are just two types of ways that we utilize them, and are currently under active investigation. So that's kind of just an overview of what some other the projects that we're doing in the lab. And now I'm going to kick it over to Anya, so a very talented graduate student who's going to tell you all of her important updates on roadmap.
Thank you, Melanie. Yeah, so hi, everyone. So my name is Anya. I'm the Biomedical Leadership Fellow, and for the past 2 years now, which is kind of crazy to say, I've been leading this project called the Roadmap. Hopefully everybody's at least heard of it, if not participated. And we're very, very excited to have gotten to this day, to Rare Disease Day, to actually launch the final deliverable, putting all of that data that we've collected over the survey, and we did 25 interviews, and put all of that data together, and created this tool. So if you want to go to the next slide, Malava. There's a little screenshot here. I tried to put in a video, but it was a little too big for the slide. So you're going to have to go to the website directly. So you can access it through everycure.org/roadmap, or you can go through the CDCN page, CDCN/roadmap, and there's a link to explore the roadmap directly there. It's an interactive tool, which has all of the data that we've collected from over 147 different rare disease organizations and their patients, loved ones, physicians, researchers. Is an enormous amount of data that we've collected. And we've synthesized it into kind of a step-by-step process of how other organizations like the CDCN can do drug repurposing. So hopefully all of you are aware of drug repurposing, of the story of Dr. Feigenbaum and sirolimus and the other drugs that we're exploring, but to become a little faster, a little better, so we can help as many people as possible with the existing drugs that are already out there, just waiting to be used to help rare disease patients. So this tool is available. It's a step-by-step process. You can click around, has a lot of information. You can go in the next slide, I think. And there's also, next slide, yeah. And there's also a data explorer. So all of the data that we've collected has this portal where you can click around, you can see the patient data. It's all aggregated, so it's all of the patients that participated. It's not specific to the CDCN or specific to any one particular patient, but you can explore the different insights of how other rare disease patients have kind of experienced drug repurposing, how is their access to drugs, and learn all of our insights that we've collected over the past 2 years. And they're also interactive, so it's kind of a fun tool. And I think Sarah is actually on. I don't know if she's has her camera on, but shout out to Sarah Bolden, who's been really great help in building out this site and all the interactivity. So great shout out to her, and thank you. And I think that's really it. I'm really excited to be launching this today. I really would love for you guys to check it out. It's not accessible on mobile, so you have to be on a computer and you have to be full screen because it is interactive and kind of a complex website. But let us know what you think. If it's, hopefully it'll be helpful for a lot of rare diseases and a lot of insights for different types of people from all kinds of organizations all around the US and around the world. And then I think I will turn it over back to Maleva and David for Ask Us Anything.
And actually, before we open it up for chatting and questions, I just wanted to say thank you very much to Michael and Melanie. I know that they're working super hard, at the CSTL, driving forward our research. They definitely can't do what they're doing without our community of patients who are constantly contributing. I personally, and even though I do this with our team, advocating for you guys every day, I learned more things tonight than I thought that I would. And so I know that it was super awesome information to share with you guys, that you guys kind of know, like, what are we doing with the samples that you guys are sending us? What did we do with the 14 samples that we got this month? So thank you both for giving us your expertise and allowing our community to learn from what you guys are doing. And then Anya downplayed this a little bit, but a huge shout out to Anya. She spent over 2 years of her time dedicated to this project. It was not just, building a tool and doing surveys, hours upon hours upon hours from Japan, getting up at 3 a.m., going to bed at 2 a.m., staying up till 7. I mean, she was just nonstop diving into this project so that today we could launch something that hopefully changes the landscape of how organizations look at, repurposing drugs. And so thank you, Anya. Thank you, Sarah, for your amazing expertise in getting this tool across the finish line as well. It is really, really amazing. I'm super thankful that you were on our team, and I'm really happy to celebrate you tonight and all the accomplishments that we are posting about now. And I think David has wrapped up his interview, spreading more awareness about Every Cure and the CDCN and the work that everybody's doing. So David, if you wanted to say anything before we open it up to everybody, and then we can, I'll stop sharing so we can see everybody's faces and we can continue to kind of answer questions.
Sure. Well, happy Rare Disease Day to everyone. It's so good to see so many of your faces. And even when I can't see your faces, I love seeing your names. Brings good memories and thoughts to me. So just so great to see so many of you guys on today. Just as was shared, I was trying to evangelize about rare diseases. I was just on ABC News in LA, spreading the word, the gospel about rare diseases and how, organizations and individuals need to pay more attention to rare diseases, which I'm sure you guys got some of these numbers already. If you look at any of our social media, you certainly would have heard them, but there's unfortunately about 10,000 rare diseases. Of course, our rare disease is the most special of all, but there's 10,000 others, and collectively rare diseases impact about 30 million Americans and 300 million people worldwide. And so obviously on our own, we're rare, but collectively we're unfortunately common. And it's really special to be able to spend time together with you on Rare Disease Day. It's hard to say that we're celebrating Rare Disease Day because obviously these are horrible diseases, but the idea that we can celebrate, great researchers like Mike, like Melanie, like Anya, share about the work that they're doing and we are all collectively as a community doing to take on Castleman disease, I think is really special. And I think it's worth celebrating. So thank you guys for all you do, because everything that Mike and Melanie talked about, it's not possible without medical data through Accelerate, without samples through the biobank, without really great ideas through the All-In movement that many of you all contributed your research ideas, without funding, which many of you guys are so generous with supporting. Those of you that I guess I'd say like the OG Castleman family people, maybe Shannon falls in that category. Definitely Penny. Penny's been around for a little while in our community for sure. But those of you will remember when it was just me and maybe one other person, and we were having webinars like this and doing everything we could to raise awareness and funds. And it's just, for me, so special to see the growth that we've been able to have, and to see the great scientists and team members that we have on our team that are working on this disease, all day, every day. Again, wouldn't be possible without every piece of this puzzle. So anyway, sorry for my long rambling, just to say, I love that it's Rare Disease Day. I love that we're spending time with you all on Rare Disease Day. And now we've got some time for what we call Ask Us Anything. For those of you guys that have been on these community updates, it means exactly what it sounds like. Anything that's on your mind, ask us anything. And whether it's for me, Maleva, or anyone else on our team, we'd love to chat through it. So happy Rare Disease Day, everyone.
Thanks, David. So feel free to just unmute and ask. If you don't feel comfortable talking, you can also just put it into the chat and I will read it out loud and we can address the questions that way.
I have a question.
Okay, sure.
Yes, I wanted to know how many people are in the United States that have Castleman's. Do you know that number? I'm just curious.
It's a great question. We've got a range, and the reason it's a range is because there was no unique code for Castleman's until 2017. Many of you all, or at least some of you on this call, were part of the petition that we put in for the U.S. government to make a unique code for Castleman's. So we have one, D47.Z2. It should be on everyone's medical records. So because there was never any data collected on this until 2017. So that means we don't have a lot of data, but the little data we do have suggested it's somewhere around 5,000 new diagnoses each year in the US of all forms of Castleman's. And we think that it's about a 1/3 breakdown between unicentric Castleman disease, HHV-associated MCD, and idiopathic multicentric Castleman disease. It's probably not exactly a third, but somewhere around that. And that's new diagnoses each year.
Oh, okay. Thank you.
Yeah. And, one disease we like to use as a comparator, because many people are aware of it, is a horrible rare disease called ALS. ALS also has about 5,000 new diagnoses each year, certainly has a lot more public awareness. But yes, also one of these rare diseases out of the 10,000.
Okay.
All right, well, I have a trivia question for you guys that you can throw in your answers into the chat. I'll say this and then we'll answer Sandra's. That's how many drugs do you think are approved by the FDA? How many FDA approved drugs are there? Which is basically how many drugs could be utilized by our group or by any group to potentially treat a given disease. So you can throw in your guesses into the chat, and it's a good guess. Throw in other guesses. And then in the meantime, we can answer this question.
Not letting me chat.
So Sandy's question there, are there any repurposed drugs being trialed for Castleman disease?
Yes. So we have a trial here at Penn of sirolimus, a clinical trial that Bridget and Tracy helped to lead, giving sirolimus to patients who are not responding to IL-6 blockers. So that is a repurposed drug. Sirolimus was made for kidney transplantation. Many of you all know the story, but we repurposed it for me. And I was the first patient with Castleman to receive it. And it's now been over 9 years that I've been doing great. And I see some folks on the call that I know are on sirolimus. I'm doing really well. So yes, that's one that we have in a clinical trial right now. We're looking to potentially open up another clinical trial of a drug called ruxolitinib. Ruxolitinib looks really promising in the few patients that have used it so far. The reason that we would potentially have two trials open at once is that it appears that sirolimus seems to be more effective in patients that are not quite as sick, whereas ruxolitinib may be more necessary for patients that are currently hospitalized or in ICU, for example. It's a bit stronger of a medication. And so we're interested to potentially launch a clinical trial of ruxolitinib. And then I should mention that at all times, every single one of you, even though you're not enrolled into a trial, being part of Accelerate, our registry, means that we're constantly getting data on what drugs are working for you and what drugs are working for other patients. And so we've got a big study coming out. We should be finishing up in the next couple of months that Sheila's been leading. I hope you guys all know Sheila as our valiant leader in accelerating all of clinical research. She's wrapping up a really important study based on almost 100 IMCD patients on the drugs that are most effective for IMCD in particular. Mateo, who I see on the call as well, is also wrapping up a really important study around unicentric Castleman disease. And unfortunately, the fact that surgical excision is not effective for everyone, despite the fact that our medical community, myself included, of course, I'm part of it, has been a bit slow to figure that out. We've got a study coming out on that pretty soon. So we're doing repurposing through trials, but we're also tracking drugs through Accelerate.
Is there anyone else on obinutuzumab, or am I the lone wolf
I think
here?
you're the only one. Obinutuzumab is a cool drug. It's the kind of the new version of rituximab, and it's thought to be maybe a little bit more effective and maybe a little bit safer, less side effects. And Penny, it seems like it's worked really well for you, right?
It has. Yeah, it might be something to look into for other patients.
Absolutely. It's a great point. And this is why we do these sorts of gatherings.
The rituximab seemed to lose its efficacy. Symptoms started to creep back on me with that. And it never got rid of anything completely. I've had more energy and been able to do more in the past few months than I have since I was diagnosed.
Wow.
That's wonderful.
That is amazing, Penny. I'm so happy to hear that.
David, I actually had a question sent in to me regarding if somebody has been on siltuximab for several months, doing really well, responding well to it, and then all of a sudden they're just not getting back to that 100% anymore. Is that indicative of them needing to potentially change treatment options, or is there something else potentially going on?
Yeah, it's a great question. So siltuximab, we know, works really well in about 1/3 to 1/2 of patients. And generally, if you respond early, it'll typically be pretty durable. So you don't usually see patients respond and then sort of lose response. But it may be that that drug was given with steroids, and the steroids were sort of helping boost up that maybe the siltuximab wasn't completely controlling it, and the steroids were sort of propping it up. And then once you took the steroids down, you're not having as good of a response. So that's one possibility. We certainly at the same time have seen patients where they've truly had recurrence of their disease, even despite a really good early response to siltuximab. So it's certainly possible, though less common. I think the key thing is going to see your doctor. And if your doctor isn't that experienced with Castleman disease, talk to Maleva. We can help you find a doctor who has more experience. Of course, we can never guarantee that the doctor is going to, give the answer that's, that you're necessarily looking for, or that is what you're hopeful to find. But we certainly can always make sure that we'll connect you with doctors that are experienced in the disease. And with siltuximab in particular, it may be that another drug might need to be added on top of it. It may be that you might be a good candidate for our clinical trial, because maybe one of these other drugs might actually be a better solution for you.
Awesome. Thanks, David. We had a question brought over from Facebook. Is one blood type more susceptible to the Castleman's disease than others?
It's a good question. We've not looked at blood type. Blood type is one of literally thousands of potential variables that could, make someone more or less susceptible to Castleman disease. And so we haven't specifically asked that question. It wouldn't actually be too challenging of a question for us to ask. And I would love for it to give us an answer. And whoever asked that question gets all the credit for it, because it really hasn't been on our radar. We've been thinking about a lot of the other, thousands of variables that could potentially be causative or leading to it. But no, it'd be great if it was as simple as a blood type.
I think we need to cycle back to your trivia question, because we have several things happening. 7,000, 14,000,
I love the guesses. Thank you for putting guesses in. Going out on a limb. So the answer is 3,000. There are 3,000 FDA approved drugs. And next trivia question, how many diseases do you think they're approved for? So you can put that in. So if there's 3,000 drugs, how many diseases do you think those 3,000 drugs are approved for? Hey, Gary, good to see you, my friend. Yeah, Penny coming in with a very specific answer. I like it. 3,200. It's very specific.
There's 600, 3,000.
All right. I think there's some cheating going on. These are very good answers.
I will say, Pete did admit he cheated. He's cheating here.
So I'll give the answer for this. It's also about 3,000 drugs. So 3,000 diseases. So 3,000 drugs are approved for about 3,000 diseases. Each of those diseases has on average about 3 drugs approved for it. So what you end up with is a scenario where we've got 3,000 drugs approved for 3,000 diseases, but these same drugs are approved for the same diseases. And then you've got 9,000 more diseases that don't have any approved therapies. We're lucky in Castleman's that we have an FDA approved drug, siltuximab. We fall into that small percentage, but then there's another 9,000 that don't have any approved drugs. I think we're really on the leading edge in terms of the number of drugs in our arsenal and that are utilized. I think in large part because that's been such a focus of the CDCN over the last 10 years, is to say, as soon as we learn something about the disease, let's see if a drug's going to work against it. And that's a bit unusual in the medical community. But I think that it's just really been the heart and soul of the CDCN is helping patients. And we're not doing research to do research. It's to help people. And so, I think that from Sheila's paper, there's probably a couple dozen drugs that have been used at least a few times. And probably half of those are used quite often. So though we only have one FDA approved drug, we've got 10 plus drugs that are used quite often. And I see Gary is referencing the fact that we're definitely on a lot of drugs. Certainly act like we're on a lot of drugs, but the good type.
One 47 different drugs were found in our data.
It's crazy. It's amazing.
Quick question. The drug repurposing is very interesting. I am curious because getting a drug approved takes a lot of time, a lot of money. It's tedious, right? Maybe if a drug's already approved for one thing and it's deemed safe for people, in some capacity safe for people, is there anything that makes it faster for being looked for the repurposing to get it fast tracked to be able to skip some of those steps to see whether or not it's safe?
Yeah, it's such a great question. You're spot on. So yeah, what's pretty cool is, I can share drugs that, that you've probably taken and that I've certainly taken. So we'll use rituximab as an example. So rituximab is not approved for Castleman disease. It is approved for a number of lymphomas. But probably half the people on this call receive rituximab at some point in their disease course. And that's because there's some, early evidence that it might be helpful for some patients. And so it's not approved for Castleman's. It's never going to get approved for Castleman's, but it's accessible to just about every Castleman's patient. And so one of the cool things about repurposing is that once a drug is approved for its first thing, it can be prescribed for anything else a doctor wants to prescribe it for. And in this case, rituximab, is used like, it's called vitamin R because it's given out so often in oncology. It's like sort of the drug that's given out the most. And so that's kind of an extreme example, but sirolimus obviously was not made for Castleman's. And so point is we can get it to patients pretty easily when you have a horrible disease like Castleman's, where we need medications to save lives. Generally, the system works. To your point, the system doesn't totally work because if you don't end up getting that drug to get another approval for Castleman's, like if you just leave rituximab is just approved for lymphoma and you never do the studies to prove that, to get it now approved for Castleman's, you're at risk that maybe an insurance company won't pay for it because they'll say, hey, it's approved for lymphoma, why are you using it for Castleman's? But the reality is that that's a lot less of an issue than you think it is, than you think it would be. It's a lot more about what's the evidence that it's going to be helpful for Castleman's and how bad's the disease. So if you got a bad disease, and you've got a decent amount of evidence, then generally, and it's rare, then generally the insurance companies will pay for it. But I'm sure there's people on this call that have struggled with insurance companies to pay for their medication. And it requires, really the big thing is finding a doctor who's willing to write a bunch of letters and make a bunch of phone calls.
Absolutely. David, there's a question posted in the chat. Are drug companies looking to fund this type of work with repurposing, which could see their pharmaceuticals used by more patients?
So unfortunately not. We're particularly focused on the about 90% of drugs that are already generic. So out of those 30 or 3,000 drugs that we mentioned, almost 90% of them are generic. And generic drugs by definition are going to receive a lot less attention from a drug company than a drug that's on patent, right? Because no one really makes any money off of generic drugs. And so we're really focused on those drugs that are already generic because no one's doing any research on them. Sirolimus, my drug, is an example of generic drug company that owns it or owned it. No interest whatsoever in figuring out new uses for that drug. But we, through the CDCN, made that discovery. We're making sure patients get access to it. So that's where we really want to lean in, or the places where like no one else is working on it. And that's where we want to lean in, which is kind of tough because Gary, I'm sure you're thinking, well, maybe they would, put funds towards it like you put in your chat. But since they're not going to benefit from it, it's hard to make an argument. But we're certain that certainly is not stopping us from asking. We're definitely asking. And I will share, the drug ruxolitinib, we're pretty optimistic that drug company may want to support our trial because it's a fairly small market drug. It's not used by that many people. And so maybe they would be interested in seeing it used in more ways.
Another question that was in the chat is, has there been any correlation between Castleman disease and military chemical warfare? Certain patients, doctors kind of leaning more towards the fact that this person's Castleman disease kind of stemmed from their time in deployments and kind of wants to know if there's any correlation there.
Well, first off, Alexander, I'm so sorry that you've sounds like in addition to your Castleman's, it sounds like you've really had a lot in your life that you've had to go through. So we have not seen that systematically. I wouldn't say. I mean, I think Mileva, I don't think either of us have observed that there's maybe a larger military presence within our community than maybe you would expect by random chance. Mileva's family is a military family, and there certainly are a few others, but I don't think it's a huge. I know our good friend Gary as well, but I'm not sure that we've seen a real sort of enrichment or larger number of patients in our community from the military. Doesn't mean that that's not exactly what's caused it in your case, but we haven't. There's a lot of unfortunately a lot of people in the military that have been exposed to these things, and we haven't seen a lot of them coming to our community with a Castleman's diagnosis. Doesn't mean it's not causative, but it just means that we haven't seen that.
Awesome. Thanks, David. Two from Facebook, and just want to give the 5-minute warning. There's 5 minutes left, so I'm going to read the two from Facebook, and then if anybody else has questions, make sure to either throw them in the chat or shout them out. The internet says that herpes simplex is linked to this. How many patients have been tested for it?
Yes, yeah, so it's no one's Castleman's is linked to herpes simplex. HHV8 associated MCD, which is not herpes simplex, is herpes 1, but herpes human herpesvirus 8 is linked to Castleman's in a fraction of multicenter Castleman's disease. It's caused by HHV8 infection. I said earlier it's about a third. It's probably a lot less than a third. We see, at least in Accelerate, we see probably almost 10 IMCD patients for every one HHV8 positive MCD patient. So IMCD means that it's not, we don't know what causes it, but we know that it's not caused by a herpes virus. So yeah, I'd say maybe for every 10 IMCDs that are HHV8 negative, and also for all unicentric Castleman disease patients that are all HHV8 negative, you'll get one patient that's where the virus itself is actually causing the disease. And I should mention about 10% of the US population is exposed to HHV8 at some point in their life. And once you're exposed to it, you have it the rest of your life. So, you know, I don't know, well, 10% of people on this call, so 5 or so people, you know, have HHV8. But if you have a healthy immune system, you can control it, and it won't cause Castleman's. But unfortunately, if you have a weakened immune system, potentially because you have HIV infection or another cause that would weaken your immune system, then you're at risk of HHV8 associated MCD.
Final question from Facebook. Are there any other companies trying to make drugs for Castleman disease?
No one's working on trying to make a new drug for Castleman's. Like I said earlier, we're kind of unusual and fortunate in like the top 5% of all rare diseases that we actually had a company make a drug for our disease. Janssen Pharmaceuticals made siltuximab for Castleman's. Then it was acquired by EUSA, and then more recently acquired by Recordati. So siltuximab is on to its 3rd owner right now. Like I said, only 5% of rare diseases have a drug developed for them. So we're lucky in that sense. No other drug companies are working on drugs for Castleman's, but we are very actively working on drugs for Castleman's through the CDCN, right? So ruxolitinib is next on our pipeline after sirolimus, and then ruxolitinib, and then a few other drugs that are, I guess I'd say, lower down, but that we're excited about because of discoveries that we made in the lab. And, you know, Mike and Melanie are hot on the case of this disease. And I think that we're going to keep learning more and more. And we always ask, you know, what drugs are already FDA approved that might be able to fix this disease based on what we learn in the lab.
Awesome. Thanks, David. Any final questions from the group on Zoom with us? That just means there is time for us to do a flex picture for Rare Disease Day.
Flex picture, I love it.
I can't take credit for it. Anya reminded me. So Anya, thank you for reminding us that we need the flex picture. But for those of you who have not done this before, Shannon's doing it right now. So everybody look at Shannon and David, the warrior flex. So we're going to take 2 seconds. I'm going to count us down 1, 2, 3. I'm going to screenshot twice because there's so many of you on to make sure we get everybody's warrior fierce faces. That way we can have this to remember Rare Disease Day. Okay, ready? 3.
And in your face too.
It's in the face. 2, 1. All right, one more time. 3, 2, 1. Awesome. Thanks, guys. Thanks. Amazing.
Thanks, everyone.
And then just the last minute, and I'm going to let David obviously close us out too, but thank you all of you for being here. It is one of my favorite times to be able to bring everybody together on these community gatherings. I personally love seeing all of you. I get to connect with all of you and know your stories and chat with your family members. So for me, I just love seeing your faces. I love seeing that you're doing well. I love seeing that you guys take the time out to connect with each other. And on the, CDCN side of things, we just love, our whole team loves seeing you guys. Most of them are in the office and they don't get to interact with you guys on a regular basis. And so to have these moments where they get to see your faces, hear your stories, is super special to them as well. And it is a constant reminder to our team why we do the things that we're doing. Special shout out to us hitting 14 samples for this month. So all of you who contributed, truly thankful for that. We definitely are able to continue driving forward our life-saving research because of all of you who continue to donate samples, enroll into our natural history registry, send us lymph nodes when you have surgeries coming up. So Bridget is our fearless leader with all things sample and tissue donation. She works really hard in the office trying to get all of these things across the finish line. So thank you for helping us meet that goal. Thank you to the people who presented tonight. Your expertise is very, very, very well loved, wrong word, whatever. I appreciate it. Thank you for sharing it with our community. And I'll turn things over to David for him to close us out.
Yeah, just echo everything that Mileva just shared, and then of course a big thank you to Mileva for all that she does for this community. So big thanks to Mileva for just being amazing, and to all of you for joining us tonight. So fun to see everyone. I guess we're in February, so we're still, about 6 months out from seeing you guys in person for the Patient Summit, but really hope that many of you all can make it to Philadelphia. We're revamping things this year to make it, as patient-centric and as fun as it can possibly be, and make sure it's a really good couple of days for everyone. So
It's a great
It's
time.
March for Peter.
Yeah,
And
that's right. It is March for Peter. That's right.
actually, it's March for a couple people on this call. So happy March.
Oh, it's amazing. Yeah. Well, thank you guys all so much. Maleva, thank you. And big thank you to our whole team from the CDCN and from the Castle, who are on late at night after working long days every day for this disease. So big thank you to them as well. Thanks, everyone.
Thank you.
Have a great
Happy
night.
Rare Disease
Thank
Day.
you.